Hello everyone, welcome to Molecura. Today we're going to talk about anaerobic respiration and why it is important to keep our cells alive and how it can lead to pathology. So now we want to talk about what happens during anaerobic respiration. Anaerobic means that there is no oxygen and the cell cannot proceed with oxidative phosphorylation. To understand this better, it's a good idea to take one step back and talk about what happens in aerobic respiration. We have already talked about this in the video of overview of oxidative phosphorylation. I'm going to briefly talk about it here because it's going to be very important. So it's a good idea that when we are looking at this, we start from the end and make our way back to glycolysis. Many textbooks or educational contents start from glycolysis and make their way up to electron transport chain. What we are going to do is going to be backwards. Now, what happens in electron transport chain is that we are having an enzyme here called ATP synthase. What ATP synthase is doing is taking ADP, it's putting a phosphate on it and it's making ATP, which is we're going to use as energy. In this process, for each 1 mole of glucose that enters the oxidative phosphorylation cycle, we have around 30 ATPs getting generated. Now, how does ATP synthase do this? By pumping protons. The protons gets pumped through ATP synthase and ATP gets generated. Where do these protons come from? They come from the energy that the proteins in the electron transport chain are using by transferring electrons. These electrons get transferred through these proteins in form of NADH and FADH2. Now, the last acceptor of electron in electron transport chain is going to be oxygen. So oxygen is going to be our last electron acceptor in electron transport chain. If oxygen is not available, none of this is going to happen. The next thing to answer is that where this FADH2 and NADH are coming from. They are coming from the TCA cycle. What kickstarts the TCA cycle? The reaction of oxaloacetate with acetyl coenzyme A to produce citrate will kick off the TCA cycle, which will end up putting electrons on NAD plus and FAD to make NADH and FADH2. Where does this acetyl coenzyme A come from? The acetyl coenzyme A comes from pyruvate. So we have our pyruvate here. This is our pyruvate. Our pyruvate is reacting through the action of an enzyme that we already talked about. It's called Lactate Dehydrogenase and it's making acetyl coenzyme A. This pyruvate is coming from glycolysis. This is also our pyruvate here. Where does this pyruvate come from? This pyruvate is coming from glycolysis. And what we have here is our glucose. Our glucose is through glycolysis giving us 2 mole of pyruvate. So what is happening here is that our glycolysis is making pyruvate. Our pyruvate is feeding acetyl coenzyme A, which makes oxidative phosphorylation happen. Now in glycolysis itself, we are going to have production of a very small amount of ATP. And the net amount of ATP that gets produced here is going to be 2 ATPs. So here we have our ADP getting converted to 2 ATP. A small amount compared to the 30 that we have here, but nevertheless we are going to have 2 ATP production here. So as we talked about in glycolysis, there are 2 reactions which require NAD+. And these 2 reactions are going to generate 2 NADH. So there are 2 reactions which each require 1 NAD+, leaving us with 2 NAD+, getting converted to 2 NADH. So when oxygen is not available, to be the last electron acceptor here, these electrons are going to get accumulated here. So this is not going forward because there is no oxygen to accept these electrons. So these electrons are getting piled up here. They will go back and they will eventually stop the TCA cycle as well. When the TCA cycle gets stopped, PDH gets stopped as well. So we don't have any more acetyl coenzyme A. So as we can see, none of this is happening when oxygen is not there. So how does the body make ATP? As we said, the majority of our ATP is coming from ATP synthase here, which is around 30 ATP. But this is not happening also. All of this have gone away. So the body needs to find a way to keep things running until oxygen comes back. And this is what happens during anaerobic respiration. In anaerobic respiration, we are going to need glycolysis to keep happening. Why? Because it's generating ATP. Although this is a very small amount of ATP, but something is better than nothing. So the cells need to keep glycolysis from going on to make a small amount of ATP and wait for oxygen to somehow come back and resume all of these activities. Now, to keep glycolysis going, you are going to need one thing, which is very important for glycolysis to go forward, and that is our NAD+. At some stage, glycolysis have gone on so many times that we have already ran out of NAD+. So the body needs to find a way to regenerate this NAD+ to keep glycolysis happening. And how does the body do this is through anaerobic respiration. So what happens, the pyruvate that we have here is going to get converted to lactic acid, and the conversion of pyruvate to lactic acid is going to regenerate the NAD+ for us. So the reaction that happens here is going to be pyruvate getting converted to what we have here is lactate. So this happens through action of an enzyme that we call lactate dehydrogenase. The important point of this enzyme is that's going to use NADH to regenerate NAD+. We said for 1 mole of glucose that's entering glycolysis, we are going to have 2 moles of pyruvate. So here 2 moles of pyruvate are getting converted to 2 moles of lactate. Thereby, they are taking 2 NADH and they are making 2 NAD+. By this method, they are going to bypass the TCA cycle and the electron transport chain because we don't have oxygen, and they will keep generating this 2 ATP over and over again until oxygen comes back somehow. But this comes at an expense, and that is accumulation of lactate in the cell. And the cell can handle lactate to some level, but after some time it's going to be problematic. Some cells in our body, like the red blood cells, lack mitochondria. So this is not happening in the red blood cells at all, because the TCA cycle and the electron transport chain are happening in the mitochondria. In the red blood cells, this is the main way to make energy. So that's why when we have a hemolysis in the body, we can measure lactate dehydrogenase level, because lactate dehydrogenase are present in large numbers in the RBC. And after hemolysis, we will be able to see an increase in LDH in the blood. Another example of a normal situation is going to be a skeletal muscle during intense exercise. During intense exercise, a skeletal muscle is going to abandon the TCA cycle and the electron transport chain and stick to the anaerobic respiration. During this, LDH is going to be very active and lactate starts to accumulate in the muscles. That's why you will get muscle cramps after an intense exercise, because lactate, which is acidic, is getting accumulated in your muscle cells. But in other situations, this could be a result of a pathologic process, and that is when oxygen is pathologically not available. So what kind of pathologies are we talking about? So one of the notable examples is going to be ischemia. During ischemia, we don't have any oxygen because the blood vessel is blocked and it's not able to perfuse the tissue with blood. So we are going to have a lack of oxygen. When we have a lack of oxygen, this is not happening. The electrons are not able to give the final electron to oxygen. So this is stopping. This is stopping. The TCA cycle will halt. The acetyl coenzyme 8 doesn't get produced, and pyruvate is forced to go towards lactate production to regenerate this NAD plus for glycolysis to go on and make a small amount of ATP to keep the cell alive. So during ischemia, the intense pain that happens is because oxidative phosphorylation is not happening and lactate is getting built up. The cells can tolerate lactate to a certain level, and after that, necrosis is going to happen. Another example is going to be hypoperfusion. Now, ischemia is going to be hypoperfusion because blood is not reaching the tissue, but hypoperfusion could happen from shock. Okay, so this was one example of oxygen not being there. Another example is shock, septic shock, for example. But any type of shock can result in this. So shock is going to be another thing. Another example is carbon monoxide poisoning. Carbon monoxide is going to get attached to hemoglobin and it will prevent oxygen from attaching to it. So the blood flow and the perfusion is okay, but there is no oxygen on the hemoglobin to go to the tissue. So carbon monoxide is also stopping oxygen from getting to the tissue. And all of these are going to lead eventually to lactic acidosis. Another example is going to be mutation in the mitochondrial DNA, which is responsible for making these proteins in the electron transport chain. So these proteins are getting made in the electron transport chain through the DNA that is available in the mitochondria itself, and that's what we call the mitochondrial DNA. If that mitochondrial DNA is mutated, we are going to have decreased amount of these proteins, or we are going to have defective proteins in the electron transport chain. Electrons don't get transported properly, so even when oxygen is available, this electron transport is not happening, and this will lead to lactic acidosis. We are going to be talking more about these proteins when we talk about the electron transport chain in more details. So there are situations where we can measure the amount of lactate dehydrogenase and the amount of lactate in the blood to be able to diagnose a condition or the severity of that condition better. I've already talked about an example which in hemolysis of the RBC we can measure the amount of lactate dehydrogenase. On a condition such as sepsis, we can measure the lactic acid level because of tissue hypoperfusion, and that can be an early sign of a septic shock. Lactic acidosis can be seen in several conditions. We have already talked about the situations in which we have severe hypoxia. We have already talked about situations which we have mitochondrial dysfunction. We can talk about situations where certain medications like cyanides or salicylates are causing disruptions in the electron transport chain, which we will talk about this in more details in future. We can talk about situations where metformin is resulting in lactic acidosis. There are many more examples of this situation which you can think about next time that you are studying pathology and try to link the biochemistry of lactic acidosis with the pathology that is happening, because a great deal of pathologies that are happening in the body is because of oxidative phosphorylation getting stopped. It is important to understand that eukaryotic life is dependent on the mitochondria to go ahead. Mitochondria is the organelle that's going to provide the body with the energy to make multicellular life possible. Any disruption in this process is going to result in pathology. Although there are cells that in normal conditions are using anaerobic respiration, and this is a normal adaptation that our body has to situations where oxygen is not available or when the electron transport chain has stopped. However, we said that when the amount of NADH is increasing, it's going to be inhibiting our pyruvate dehydrogenase. So when the mitochondria is knocked out, it means that the electron transport chain is not happening, the TCA cycle is not happening, there is nowhere for this NADH to go. So this NADH in turn are going to be inhibiting our pyruvate dehydrogenase. They're going to come here and they're going to inhibit this pyruvate dehydrogenase. So they are practically telling the body that this is not happening. So you will need to find a way to keep the energy level high enough for the cell to stay alive. And our body's way of doing that is by making glycolysis go ahead over and over again. But because glycolysis is going to need NAD plus to go ahead, it needs a way to regenerate this NAD plus. And the regeneration of the NAD plus through NADH is going to happen by the action of lactate dehydrogenase, which is going to take the pyruvate, convert it into lactate, and in process convert our NADH back to NAD plus for this to go ahead. This can be a good adaptation for our body to keep the cell alive as much as possible. But at one point, lactic acid buildup is going to overwhelm the cell's adaptive mechanisms and is going to result in necrosis and cell death, which we will talk about more in the future. This was our video on anaerobic respiration. Before we jump into TCA cycle and talk more about what happens during TCA cycle, we will have two short videos talking about fructose and galactose metabolism and the diseases that are associated with them. If you enjoy our content and this is helpful for you, please make sure to subscribe to our channel and explore more videos on our biochemistry series.